Thank you everyone for going through and listening to this presentation, and we can go ahead and open for questions for anyone. You're welcome to use the chat or to unmute yourself and present any questions. And I think some things that we can just review too that are updated at least from our procedures, so that you know if you are interested at any point in starting a gene curation expert panel or participating in a gene curation expert panel with ClinGen, performing the lumping and splitting is really part of this whole group's, what would be under their purview. I've put in the chat what is the link to the lumping and splitting working group. From here, you can see the guidelines that we have drawn up here, again, which was reviewing exactly what we went over today. What's nice is that you can also look at the pre-curation information. This will give you kind of a template, but I'll also go through that we have an actual system that we can capture this pre-curation, but it also will present an example for you. So you can see the types of information that you want to capture. And that's updated. And we also have a tutorial here similar to what you have heard today. So certainly you can go back and look through those. What's really nice is that recently we have created what we call our gene tracking system, which helps to support our gene curations as well as the gene curation interface. Here it gives our curators the option to be able to enter in their gene of interest and also indicate which phenotypes are part of the curation. What's nice here too is that when you go through this, as if you become part of a gene curation expert panel, once you enter in this information on genes, it will tell you whether or not other groups have interest in this particular gene curation themselves. So you can go through and be able to collaborate and communicate with those individuals. But as you'll see, you're able to pick out phenotypes and assert which ones are going to be part of your curation. We do have plans in the future to publish these to our website so that you will be able to see what the lumping and splitting decisions were for each gene disease relationship. And that is also going to help us to harmonize with other entities such as Online Mendelian Inheritance in MIN or OMIM, as well as Mondo, where we use our disease entities from. Are there any questions from anyone?
Hi, thank you very much. Mine is not a question, but because I saw that on one of your slides, you showed a table of genes, and I see the Cavelin tree. So it's just a practical question because it is reported to be mostly dominant, but also some recessive cases. So we have a family, we did exome sequencing, and we found heterozygote variants, 2 in the same gene, but they were reported to be SNPs. How can we best address this question? Like finding if those reported SNPs are reported on the same individual.
That's a good question. Certainly coming through when we think of lumping and splitting too, like, oh, all of a sudden, are we getting a dosage effect, right? It's known as the dominant, as you said. There are potentially 2 different variations in CAV3. Certainly this is a little bit of going into, as you said, are they actually on opposite alleles? So that could be going back to your patient, if possible, to test parents, right? If they're still alive, to show that they have inherited an allele from each parent. Sometimes that's going back through to figure out if there's no parents, are the siblings also, do any of them have separate variations, right? We would assume that if they were 2 variants in cis, that there's the potential that another sibling would also have those same 2 variants. Now, of course, you'd have to still distinguish if they're on different alleles. But as far as lumping and splitting too, we have to also assess whether or not there's a dosage dependent effect once you figure out that, yes, they're actually in trans and they're not in cis. And we still have that ability. The most well-known example of a dosage dependent effect is LDLR, right? We know that it's associated with both dominant familial hypercholesterolemia and also recessive, which is more severe and early onset. So in this case, if you reported this and we went to review it, we'd really want to see those in trans and whether or not any of the phenotypes overlap here and whether those variants also overlap in the dominant disease. Is it representing, a dosage dependent effect of a dominant disorder, in which case we would call that semi-dominant, a semi-dominant mode of inheritance, where it really explains that dosage. In those cases, we can lump those together, and we had, with LDLR, and we do have a new semi-dominant mode of inheritance, and we have the ability in our GCI now to curate both on dominant and recessive alleles in the same curation so that we can represent the full spectrum. But also it's a great question because, with the ataxia telangiectasia, the ATM example, we know that they have some of the similar phenotypic features. They have cancer that overlap between both of them. But again, in that dominant disorder, when we think about the breast cancer susceptibility here, they are not at risk of developing the ataxia and or the telangiectasias that are developing, right? And so this is what really begins to distinguish them as different diseases for mode of inheritance. So that's a really great question. I hope I answered some of what you had asked, but thank you so much.
Yeah, sure. Thank you.
And I just want to point out too, depending on how many individuals here are clinicians versus geneticists, a big thing that comes about is, we do appreciate and understand, again, that individuals are coming with you as a phenotype, and that's how they're being diagnosed. So, and taking some of these curations, and I have a couple of examples here of different genes. This is RET for multiple endocrine neoplasia. This is not in any way to say that, if we lump some of these together, that we're suggesting clinicians diagnose individuals with this. That's not what we're asking, and it's in no way to disrespect anybody's job as a physician. You certainly have to treat the person for the diagnosis, but what it really helps is for us to broaden our mind to think about variation, whether we're looking at pleiotropism or heterogeneity, which we've now found with looking at underlying genetic variants in disease. It's really just to bring together that information. And it could be quite important too, if we're only looking, for instance, just in heart, because we're a cardiologist for a heart-related phenotype, we may miss the fact that there's some skin or hair-related phenotypes. And we know that there are, very well-established genes that have different, heart, skin, and hair-related phenotypes. DSP is one of them. And so it's really important when we consider that we curate DSP for that full syndrome, because that will also bring attention to anyone coming to ClinGen's website, like, oh, I have someone with a DSP variation. They do have dilated cardiomyopathy. I may have missed that they had really curly hair, and I didn't really pay attention about whether they had skin that was breaking or not. Maybe I should go reassess them for that. We've also had very disparate types of clinical phenotypes present, where we can have a hematological phenotype that's also associated, potentially with a hearing loss or something, where you just wouldn't really necessarily, as a clinician who's looking at hearing loss, think about that blood disorder. So it's really just to make sure we're representing the strength of those relationships most appropriately. And also, we don't want to confuse our stakeholder. One of the genes that we had in MEN1 at first is one of the key ones that we had gone through to look at. Multiple endocrine neoplasia type 1 is associated with this phenotype. However, at one point, it had a list of different genes, and it included pheochromocytoma, which is a phenotypic feature that's very rare but can present in individuals with MEN1. And that was split and had a different classification of moderate versus what would be definitive for here. And that might confuse individuals, especially patients, if we think they may come to our lumping and splitting. They may say, oh, am I at a moderate risk for developing it? And that's not exactly what our curations are doing, right? It's really just saying how much evidence is representative in the literature for a specific disease. And so that's what really gave us the impetus to do lumping and splitting. That's not to say for a group, if you want to call out that it's a very rare phenotype, you could by just presenting the amount of individuals in the literature that have been noted to have this phenotype, but it wouldn't require a formal curation with the classification at this point.
So yeah, I have a question.
Yes.
What about assessment of digenic combinations in case of oligogenic inheritance or polygenic inheritance?
That's a really good question. Right now for the lumping and splitting, it really is confined to Mendelian or single gene disease entities, but we do hope to move out from both our gene curations into the polygenic or digenic. Certainly there are some, very standard digenic genes that come together. We would also want to, when it is known to curate, right, that these two genes are associated with this disease entity. And that would be really important. So I think it will evolve as we move through, but at this point, it's just for those single gene disease entities.
Okay.
Courtney, we have a question in the chat. Is or are there standards for experimental validation of the association between genes and variants and disease of interest during curation?
Oh, that's a great question. Certainly, we take into effect this experimental validation. ClinGen has just gone through, even when we think of variant interpretation, to look at what are the appropriate guidelines for a functional assay. And this is really to include whether or not benign variations were tested versus known pathogenic. And that's really to validate that experimental assay. And that becomes really important for gene curation too. We do have to do an amount of work ourselves to indicate with a functional assay whether or not it's really appropriate. Certainly, if you know the molecular mechanism of the disease, this often comes from experimental validation, and that's what's helpful. We can distinguish disease entities by those different experimental assays. But there's an amount of it where we really have to involve ourselves into figuring out that molecular mechanism, especially because we know literature is biased, that it will tend to say something's novel. So oftentimes we'll come to genes where one says it's a loss of function, another a gain of function. But one way we meet this is that if they do have different variations in the gene, certainly we would encourage individuals to go look at NOMAD if they haven't already indicated where they're pathogenic or benign. This is something that was really important in the intellectual disability and autism group when we looked at electrophysiology. We were able to find a lot of variants they thought were pathogenic. When we looked at the general population, were too high of a frequency to be considered pathogenic. That already meant that those were benign, and we could compare all the other variants to that and really give us an idea of molecular mechanism. So I would definitely encourage you to look at different population frequencies also within your own patients and your own data that you have in your databases to really help you kind of distinguish the lumping and splitting. And certainly if you find a phenotypic feature where all those variants seem to be too high of a frequency to be pathogenic, that's where we would encourage splitting off that phenotype to dispute it. That would be really important because that gets this off of different websites like OMIM, but it really gets out to the rest of the world that, variation in this gene for this disease maybe shouldn't be returned to individuals as a result. So I hope that answers your question. It was really great.
Thanks, Courtney. I also stuck in the chat Shannon's paper from your team about functional assay validation. So another question we have, would you please elaborate more on how to join a GSEP?
Oh, great. Yes. So if you want to join a gene curation expert panel, you can visit the ClinGen website here. You can go underneath our working groups and you would go to the clinical domain working group. Underneath this particular one, you do have to scroll down a little bit, but each one of our pages has a separate area for documents. You can have the guidelines for applying for a variant or gene curation expert panel status. This is really if you want to start a new gene curation expert panel and build it together. You can go to this particular document and then you can begin to download those expert panel applications for gene. This shows you the quick guide of everything that you'll need to have. And then it gives you the contact that you give when you want to submit this application. Now, if any of you want to actually join one of our gene curation expert panels that we have at this time, that's listed under expert panels here. What we do have is we have our community curation working group. Through this working group, we give you training for the gene curation, and we then will help to place you on to expert panels that are accepting. Our website shows you those gene curation expert panels and any of our other working groups that are accepting at the time. You can reach our survey through several different tabs here that will take you to our community curation database. and you can sign up. If you want to join an expert panel, we would encourage you to pick the comprehensive curation effort, and you'll be able to select gene as your primary prioritization, and then you'd be able to pick the expert panel you would like as well. So those are two different mechanisms to come in to ClinGen, whether you want to come to one that's already curating, or if you want to come to one, develop one yourself.
Great, thanks, Courtney. Everyone feel free to keep adding questions in the chat. Those are really great questions. I was wondering maybe if you should show them, just because you showed them the great gene tracker, but right now it's not publicly available on the website. So maybe if you could show them one or two curations and like kind of in the summary where they would see a little bit of the lumping and splitting decisions.
Sure.
Publicly, at least at this point until the tracker information is available, if they were to browse genes on their own.
Yep. So we can pull up some different genes here. Let's see if we have our Lucky Men one. So when you come to our website, you can come to the search toolbar. You can type in by a gene of interest using an HT&C approved symbol. You can also type in a Mondo ID or a phenotype description and be able to pull down several different examples there. You would go to that little green button for report to pull back the report for each of our gene disease validity classification summaries. And where Marina is talking about is within this evidence summary section, you may find for different genes where they have lumping and splitting assertions for them. So this is one area where you can really see whether we've lumped or split the curations. And so I should probably pick some other curations that we have. Oh, I think I put in. Having to remember all the gene names is sometimes a big change here, and I'm not doing really good today. Marina, do you have an example?
Are you looking for one that's lumped or split?
Either one. So here, you know, again, we have neurofibromatosis type 2, and we can go in with these evidence summaries just to indicate. So here's the lumping and splitting kind of statement here. We'll say that it's associated with several different entities at the time, but that they were not part of a separate curation, or whether or not they were lumped for a full curation. So you will see these statements in the interim as we work to get more information from the gene tracking system on the ClinGen website.
Yep, that's great. Thanks for thinking of examples on the fly.
No problem.
Does anyone have any other questions? Yeah, in the chat or verbally. I think we have a few more minutes left.
But what becomes really nice if I add a little bit more is that, you know, regardless when you come into the gene curation, really looking at the different genes, as we said on OMIM, is where we start off. You know, here's an example of HBB, which we know is associated with sickle cell anemia. And so we would just encourage groups that while we know that this would be a definitive relationship for sickle cell anemia, is that not to just do pre-curation over, you know, the particular phenotypes of interest to your group, but really looking at all of them. It's seeing that information that you have. As we had indicated, ClinVar can become a really useful site to look at this information. If you wanted to look over a specific gene of interest, I can pick one here that I know is associated with 10 or 11 phenotypes. You can search ClinVar here. And what's nice and what I typically begin to do to help myself out is I'll look at pathogenic mutations within ClinVar. Obviously, this is dependent on, you know, each individual who has asserted this, their criteria, rather than just a ClinGen mediated, but it is still a really good example. We can go to multiple submitters, right? Because then we can kind of compare what each person has picked, and then looking at the single gene. So here I've restricted myself from maybe 600 to 1,000 variants to 10 variants. But what you can begin to do is by looking at the conditions here, get an idea of whether or not they think this is a part of a spectrum or if it's really an isolated gene, and looking at those variants. It's a really good place to go when you think about data that's already been curated ahead of time for us. And this is what helps to make that process of pre-curation a little bit more expedited than going through each of the details. When you come down here, you can begin to see and appreciate that we're looking at evidence for Charcot-Marie-Tooth syndrome, which is a neuropathy, but we also know that this has been associated, this variant, in skeletal dysplasia, and that's even noted here in this evidence summary, and it does provide other PMIDs. So as a curator, this really helps us to come through and see what people have already found, lets us go back and look at this literature and really assess for ourselves if this is accurate and whether we can use this information. And again, with other individuals, we can see that we're getting in those skeletal dysplasias. So on a high throughput level, this may tip me off as a curator to say, wow, this may really be a lump for a neuropathy and a skeletal dysplasia. But obviously, as each of those expert panels come together, they can collaborate and discuss whether that makes sense, clinically, as well as the rest of the information that we assess from this.
Yeah, if I can add something onto this.
Yeah.
Because this particular gene was found through my PhD thesis. So I was working on 2 of the families. I think the difference we found here is that the mutation on the neuropathy lies on a different phase of a particular domain, and then on a concave phase of the domain, and for the skeletal dysplasia is on the other side of the protein. So we were arguing that this makes these 2 phenotypes different. Maybe interactors are different for the neuropathy compared to the skeletal dysplasia.
Yeah, that's one really great thing to look at, right? And that's certainly part of what we do in the curations and look through it. And you're right, there are some that have definitely different domains. We can go through and look at some different reviews. So I'll pull up just as an example, part of our pre-curation slides for this. We do have each group, our CMT group or Charcot-Marie-Tooth, as well as our skeletal dysplasias looking at this one. But just as you said, it could be in different domains. This paper actually has a review that goes through a lot of the different variants in the domain. So this would be another really great thing that pre-curation does, is that as you begin to look more and more into that variant and finding papers that can tell us, we can see that actually when we look at skeletal dysplasia and neuropathy over what's been published in the literature so far, it really traverses all of the domains within TRVP4. And we do know that there are some variants that overlap in both of those domains. Yeah, and so, these are things that they just have to, as we come together, begin to dissect a little bit of, is this one whole spectrum? Are there individuals who actually present with both skeletal dysplasia and a neuropathy, which is coming and emerging out more and more? But they may not, and that might be another reason, right, to split these between a dysplasia and a neuropathy. So those are really great questions and things to point out. But this is just to give you an idea of that pre-curation information we give anyways. We always ask everyone to give a background about the basic gene itself. This gives us an understanding of what its function is and why it may be participating in multiple clinical domains. If we think about, for instance, ion channels, we know that they control the brain, they control the heart, they can work in muscle. And so we can kind of predict that there could be at some instance a variation in an ion channel that's present in all of those organs that might create a actual syndrome, right? That would be a type of channelopathy. We also encourage individuals to look at OMIM and Orphanet as well as Mondo just to see what's been asserted in the literature. This sets off our groups to really get their brain into how complex this gene is. Then obviously, if there is a review or a paper that goes through to outline all of these different variants in the genes, this is where we start hitting into that molecular mechanism, right? The first 3 slides we're looking at assertion, now we're looking at molecular mechanism. Are there different variants that cluster in different domains that can tell us there's a different mechanism for one entity versus another? Do we see that there's a heterogeneous representation amongst all of the domains? We then move into phenotype and see, are there things that are starting to overlap in the phenotype? Can we see that there's, neuropathy and several things where SMA and the neuropathy may be part of the spectrum, right? We can also look at those skeletal dysplasias, and do we start seeing similar phenotypes that have been shown through each of them? And then, heading up on the rest of the mechanisms that we have, or the inheritance pattern, right? And the inheritance pattern for those genes were all autosomal dominant. So we just bring it back around to the groups to kind of figure out those 4 questions and whether we have that lump or split. What's really important at the end of the day is we need to get a Mondo term. And it's really important because, as you learned from Aaron Briggs on the first day, is that to create a gene disease mode of inheritance record in our gene curation interface, we need this all important Mondo ID. And we want it to reflect the disease entity that we're curating. Sometimes that entity may already be within Mondo, and that's great. Other times we need to update it because it's not exactly how, our experts want the terminology presented. Other times we actually have to develop new nomenclature. And that's a really exciting part of the work that we do, is that gives the chance for our groups to be able to make those new nomenclatures. I'll show you a little bit of an example of some of those different disease entities that our groups have worked on within Mondo. One of them was the complex neurodevelopmental disorder. Our neurodevelopmental disorder, CDWG, had realized that they needed a little bit more of a broader phenotype. And so they worked to develop this whole Mondo ID. It was new to them. They indicated what the definition was. It was associated with their particular domain. And they were able to go and have a reorganization of what groups of phenotypes or diseases that were underneath this parental term. So this is also part of the work that we do.
One last question, Courtney, since we only have 4 minutes. One participant was wondering about curation of non-coding genes.
Oh, that's good. Again, because this is a Mendelian, we really are looking at those coding genes that are associated with disease. But, we also understand that there could be some non-coding genes, or there can be other genes that are regulating genes and diseases differently. I cannot remember the gene off the tip of my tongue, but there was one in colon cancer like this, where there was actually a variation out of the non-coding region of the gene that was affecting the promoter and how well that gene was expressed. So the variation wasn't actually in the gene, but it was associated with that gene. So that was definitely a complex example we had to go through of what gene do we actually pick, because it may be part of the promoter region, or it may be part of another gene that's having a genetic effect on the expression of another. We do, though, try to stick to just the Mendelian inheritance at this point. Clinton will be moving into looking at polygenic risk scores in the future, and we will hopefully eventually move to when we think of multiple genes. But at this point, it is just Mendelian. Thank you.
Yeah, I second that. I'm guessing you mean EpCAM in the colon cancer area.
Thank you.
We did have a MIR, I think MIR 96, a microRNA in hearing loss that we did evaluate because it followed a monogenic inheritance pattern essentially, but most groups haven't started to evaluate that yet. But it's definitely something in the future. Along with mitochondrial genes, we have a whole group that's evaluating mitochondrial genes right now.
Yeah, great. Well, thank you all so much for all of these wonderful questions. I know we have about 2 minutes left, but again, encourage you all to come see the lumping and splitting webpage that we have here. This is on clinicalgenome.org. It's underneath the working groups tab. You can go to lumping and splitting here. We've also put that in the chat. You can see the documents that we have that are already part of this guidelines, those pre-curation information in the tutorial. What's nice is that you'll also see, once you start going through gene curation training, if you all decide to join one of our currently curating expert panels, or you will form one on your own over a specific clinical domain or gene of interest, you will end up in having to go through gene training. And this is also part of the required training parts. So you'll see here under standard operating procedure, this is really considered one of the SOPs for our gene disease validity to have to read those guidelines and attest to that. You can also see again, if you do want to come to volunteer, you will be given some emails to tell you to do this asynchronous or autonomous training. And then we do conduct through our gene community curation working group live GCI training sessions. Will get you involved in knowing how to enter in the information and curate into our gene curation interface. Those are held on a quarterly basis, and we are actually starting to plan our next training session for early March. So again, I really encourage you, if you want to be part of any of our gene curation expert panels that are currently operating right now, you can go to, the Clinton Community Curation. Here, you can take our survey, and no problems if you just hit the clinicalgenome.org website in the front. You can also come down and say volunteer to curate. It will take you as well to that survey. Oh, and we're going to be leaving our breakout room soon. So
I stuck that survey in the chat. I'm going to stop recording now since we'll automatically be